Good morning, good afternoon, and good evening, everyone. And welcome to the Scientist.com and STEMCELL Technologies webinar titled High Quality Mobilized Leukopaks to Enhance Your Cell and Gene Therapy Research and Development. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be your host today. We're being joined by Saba Aslam and Dr. Annie Bargsten, respectively product manager and field application scientist in primary and culture cells at STEMCELL Technologies. And with that, I'm very pleased to hand things over to the first speaker, which is Saba. So Saba, thanks so much for joining us today. And the floor is yours whenever you're ready.
Thank you for the introduction. I'm excited to talk to everyone today about how you can leverage primary cells for your cell and gene therapy and development research. First, a quick introduction to STEMCELL Technologies as a company. We are one of Canada's largest biotech companies, founded in 1993, and we support life sciences research through direct sales offices in North America, Europe, Asia Pacific region, along with all the distributor regions that are shown in the lighter gray. So depending on the primary cell product of interest, there may be regulatory restrictions that impact availability in your specific region. Mobilized Leukopaks, the focus of this webinar, are currently only available in North America, specifically US and Canada. If you want to learn more about which products are available in your region, you can browse our website for product pages, or you can reach out to your local sales representative or procurement contact. We have 30 years of experience supporting life sciences research and over a decade supplying primary blood and immune cell products. We are committed to advancing scientific discovery across many different areas. These are the major research areas supported by STEMCELL Technologies. The foundation of the company was built on the hematopoietic or heme and immune research over 30 years ago. And in addition to these core areas, we support a broad range of fields, including pluripotent stem cell biology, neurobiology, pulmonary research, and many more listed here. For this webinar, we'll specifically focus on immunology and hematopoietic research with an emphasis on primary cells and mobilized leukopaks. Now that we've set the stage, let's go over today's agenda. I'll begin with the portfolio introduction, followed by Mobilized Leukopak product intro, and then Annie will take over to cover the scientific overview, leukopak processing, and CD34 positive cell isolation. After that, I'll return to discuss culture, expansion, and differentiation media, along with analysis and cryopreservation workflows. And finally, we'll wrap up with a summary of key takeaway and some resources for you. Now that we've introduced the agenda, let's dive into our primary cell portfolio and how it supports immunology and heme research. So before diving into primary cells specifically, I wanted to just quickly introduce what our full workflow offerings look like. We offer a comprehensive range of products supporting scientists, from sourcing primary cells to cell isolation, gene editing, culture, expansion and differentiation, and analysis and cryopreservation. For this section, I'll be focusing on the cell sourcing aspect of our human primary cell product portfolio. So the primary cell portfolio can be divided based by tissue sources. We have a lot of different human primary cell products that come from peripheral blood, mobilized peripheral blood, umbilical cord, or bone marrow. The peripheral blood is also known as circulating blood, and it is widely used for immune and blood cell research. Then we have sources of the hematopoietic stem and progenitor cells, or CD34 positive cells, and these include mobilized peripheral blood, which is similar to peripheral blood, but involves a process called mobilization, which releases the CD34 positive cells from the bone marrow into the peripheral blood, so they can be circulating for a collection. Then we have cord blood that comes from umbilical cord that contains a naive population of stem and progenitor cells. And then we have bone marrow, which is collected directly from harvesting adult bone marrows. Each of these sources offers unique advantages depending on your research focus, from basic immunology to advanced therapeutic development. Understanding where primary cells come from is essential, but it's equally important to understand how we classify them. So these primary cells can be grouped into three main groupings that are shown here. We have bulk primary cells that include leukopak products and mononuclear cells, such as PBMCs that come from peripheral blood. Then we have our CD34 positive cell tissue sources. This includes the rest of the tissue sources that are mobilized peripheral blood, bone marrow, and cord blood. And then we also have isolated immune subtypes from specifically peripheral blood. And these include products such as T cells, B cells, NK cells, monocytes, neutrophils, and many more. One of the most effective ways to obtain a large quantities of these cells is through leukopaks, which are collected using a specialized donation process called leukapheresis that I'll introduce here. So the leukapheresis process is used to collect leukopaks from peripheral blood or circulating blood. And this process involves a few key steps to efficiently isolate white blood cells while ensuring donor safety. The first step is that whole blood is collected from the donor through an IV tube in one arm. And then anticoagulant ACDA is added immediately to prevent any blood clotting. The blood is then passed to the apheresis machine, which separates the white blood cells from the red blood cells and other components. Red blood cells and other components are returned into the donor to maintain balance and donor safety, whereas these white blood cells are the cells we want are collected in sterile bags and make the products either leukopaks or mobilized leukopaks. The donors providing non-mobilized peripheral blood are recallable and they can return for donation once every 1 to 2 months. And then here we look at how this differs from a mobilized leukopak. So in terms of a mobilized leukopak donation process, it follows similar steps to the leukapheresis. However, prior to leukapheresis, these donors are injected with a mobilizing agent 1 to 5 days prior to leukapheresis. While we will dive into the specific mobilizing agents later, overall, what these agents do is to stimulate the release of those CD34 positive hematopoietic stem and progenitor cells from the bone marrow into the bloodstream, leading to a higher yield of these CD34 positive cells during collection that will be then going to that bag that we'll collect for mobilized leukopaks. The rest of the process remains the same, ensuring an efficient collection of mobilized leukopaks for research and therapeutic applications. So that's how we collect our leukopaks that are the part of the bulk primary cell sources. And the advantage of these bulk cell sources is that you can get a variety of cell types from one single donor. So we have leukopaks both from peripheral blood, mobilized and non-mobilized. And they really allow you to not only get variety of cell types, but also large cell quantities from a single donor. So in the case of a fresh leukopak, for a full pack, you can get 10 billion total nucleated cells. For a half pack, 5 billion. For a quarter, 2.5 billion. And for a 10th pack, 1 billion minimum. Along with our product, we share donor characteristics such as age, gender, ethnicity, weight, and height that are listed on the certificate of analysis that is shipped with each product. But you can also select other custom capabilities that you may be interested in, whether it's HLA typing, flow cytometry panels, and many, many more. The advantage of a leukopak is that you can use these products as a standalone, or you can also use these to isolate other downstream products, such as isolated subsets, and really use a customized isolation protocol to get exactly what you need from the product. Along with the leukopaks, we also provide mononuclear cells that are isolated from the leukopaks using density gradient centrifugation. And these mononuclear cells come from all the 4 tissue sources we introduced earlier. And these cells are pre-allocated from a single donor, which really allows researchers to purchase smaller vial quantities, pre-qualify these donors or lots to ensure that they're compatible with their experiments, and then place the remaining part of the lot on hold for future use. We have a really flexible hold policy, so you can pre-screen and test your lots and donors, and then we'll hold these batches for you until you've completed your testing, so they're ready for you when you need them. These bulk products really provide researchers with large cell volumes and a large variety of cell types within one donor that can be used directly or processed further, offering flexibility for various research and therapeutic applications. Additionally, the mobilized peripheral blood, bone marrow, and cord blood serve as key CD34 positive cell sources, providing researchers with those hematopoietic stem and progenitor cells. So let's look at what these CD34 positive cells actually are. Hematopoietic stem and progenitor cells, or HSPCs, are essential for blood and immune system development. These CD34 positive cells include both hematopoietic stem cells that have long-term self-renewal capacity and progenitor cells, which are more committed to differentiation. These CD34 positive cells are multipotent, which means they can self-renew, but they can also give rise to all mature blood and immune cells. They differentiate into two main lineages, myeloid, which includes monocytes, neutrophil, granulocytes, and lymphoid, which give rise to T cells, B cells, and natural killer cells, and many more. Because of their ability to both self-renew and differentiate, HSPCs are critical for applications in transplantation, gene therapy, and disease modeling. Now we'll take a closer look at how these different sources of CD34 positive cells compare to each other. So each of the CD34 positive cell sources offer distinct advantages depending on research and therapeutic needs. These sources are presented here from the largest to smallest in terms of CD34 positive cell yield. Mobilized Leukopaks provide the highest quantity of CD34 positive cells, making them ideal for transplantation and gene therapy applications. And because mobilization mimics the process used in patients undergoing autologous cell therapies, these cells are particularly also relevant for disease modeling and therapeutic development. They also allow for donor recall and contain high number of T lymphocytes, so you can get multiple cell types from one product. And mobilized leukopaks yield more than 10 times the CD34 positive cells in terms of frequency when you compare them to normal non-mobilized leukopaks. They also provide roughly 2 to 6 times more total nucleated cells when compared to non-mobilized leukopaks. Bone marrow, on the other hand, has a lower CD34 positive cell count than mobilized leukopaks. However, it is known as a traditional transplantation source. It also offers recallable donors, and the advantage is it also contains other cell types such as mesenchymal stromal cells and other components. Cord blood, while it is the smallest source of CD34 positive cells, has superior cloning efficiencies and proliferative capacity compared to adult sources. It is also devoid of mature T cells, making it really valuable for certain applications. By understanding these differences, researchers can select the most appropriate CD34 positive cell sources for their specific studies and research needs. While CD34 positive cells can give rise to T cells, B cells, and K cells monocytes, these immune subsets can also be directly isolated from the leukopaks or peripheral blood and used directly. So I will introduce what those isolated frozen immune cell subsets look like. So we also provide these pre-isolated immune subsets, and the advantage of these is that by using pre-isolated and cultured immune cells, researchers can save valuable time in the lab because they really reduce the time needed to process and do the isolation. So the cells are ready whenever you're ready to do your cell assays. Now that you're familiar with all the different primary cell sources, let's explore the applications by their groupings. So as you can see, the applications vary widely based on the starting source and the intended use. Bulk primary cells, including PBMCs and leukopaks, can be used for flow immunotyping, high content compound screening. They can also be used to isolate those primary immune subsets. They can also be used for sequencing, disease modeling, toxicology, and in vitro vaccine development. CD34 positive cell sources can be used in many other different types of applications, such as the colony forming unit assay, which really looks at the functionality of the CD34 positive cells. They can also be expanded in ex vivo. They can be differentiated into myeloid and lymphoid cell types. They also have regenerative medicine applications. They can also be reprogrammed back into pluripotent cell cells, such as iPSCs, but you can also do humanized mice models, and they have a wide range of both cell and gene therapy and also gene therapy applications. Isolated subsets such as T cells, B cells are really critical for cell-specific assays. They can also be used for cell activation, expansion, and culturing. They can also be gene edited to do more of those cell and gene therapy applications such as CAR T. You can also use them for co-culture experiments to see how they interact with each other for a comprehensive immune system study. Collectively, these primary cells support cutting-edge advancements in cell and gene therapy research. Now that you're introduced to the primary cell portfolio, I also wanted to mention that primary cell products are held to the highest quality standards, ensuring safety and reliability. We conduct thorough viral screening using a standard panel of HIV-1, HIV-2, hepatitis B, and C for every product, with some products undergoing additional viral screening panels for even greater assurance. Each sample is validated for count, viability, and purity through quality management systems certified to ISO 13485 and ISO 9001. Detailed cell count and viability information is provided on the certificate of analysis, which is shipped with each product. The majority of our samples are collected in the US, and all of them are ethically sourced with institutional review board approved consent forms and protocols, with some collected in Europe using research ethics committee approved consent forms. These forms are readily available upon request. And to protect donor privacy, personal identifiers are removed, ensuring full anonymity. Donations are available for both research and commercial use. Ensuring high quality primary cells are just one part of the equation. Having a diverse and well characterized portfolio is equally as important. Our human primary cell portfolio spans multiple tissue sources, offering both fresh and frozen products to support a range of research and therapeutic applications. This slide shows all the key sources up top and the fresh and frozen products within each source underneath. This is just a really high level overview. Our portfolio contains over 160 unique products and more than 400 catalog numbers to incorporate all the various sizes and formats. And this only shows a small picture. For example, we also have a disease portfolio that covers over 40 diseases, including autoimmune, inflammatory, and cancer-related diseases that are coming mainly from the peripheral blood tissue source. We also provide biofluid fractions such as plasma and serum for supporting diverse research applications. And we also have custom offerings for any products that might not be available on the shelf to ensure flexibility for unique applications. So, with a clear understanding of our overall portfolio, the next section will now focus on fresh mobilized leukopaks and their unique advantages and applications. Fresh mobilized leukopaks serve as a foundation for many downstream stem cell products that we'll explore later in the presentation. But I wanted to introduce what our fresh mobilized product looks like in the beginning. This is an overview of our fresh mobilized Leukopak product offering. And I wanted to emphasize that these are only available in North America. So on the left-hand side, you can see the mobilizing agent. This includes GCSF, which is our granulocyte colony stimulating factor, Plerixafor, which can also be known as Mozobil. And then we also have a combination of both, including GCSF and Plerixafor. So after that, you can see the mobilizing regimen. So GCSF and combo are both 5-day regimens. So this involves giving the donor the mobilizing agents over the course of 5 days, whether it is the GCSF or the GCSF in combination with a Plerixafor. And then on day 5 and day 6, there's a first collection and second collection of the leukapheresis process. For both of these products, the collection options include the first collection, the second collection, or both, allowing flexibility based on your specific research needs. And then for Plerixafor, it's just a 1-day regimen, and this only involves the first collection. All of these options really provide researchers with customizable solutions to support a wide range of applications. Along with your product, we also ensure that you get all the information needed. And this is listed on your certificate of analysis. And this includes our viral screening panel, including our standard HIV 1, 2, Hep B, and C. But also additional panel that is included with each mobilized leukopak is HTLV 1 and 2, syphilis, and also the West Nile virus. High resolution HLA typing is also available, covering HLA A, B, C, DR, DQ, and DP. Additionally, donor recall is possible for both mobilized and non-mobilized products from the same donor. However, because of the nature of mobilization, the recall timeframes can be quite a lot longer than some of our non-mobilized products. The donor demographics range from 18 to 40 years old, ensuring consistency in study populations. And along with all of this information on the CFA, we also provide the cell count and viability and the volume. We also provide the purity for CD34 positive cells frequency. This can be gated using ICH protocols, and both ICH gating and quantification is available upon request for those who require the standardization method. In addition to CD34 positive cells, we also do the flow panel on CD45 positive single viable cells. We provide the flow plots for T cells, including CD4 positive and CD8 positive, the monocytes, B cells, and NK cells. The mobilized leukopaks are shipped in an overnight delivery in NanoCool Universal Long Haul Shipper, and these control the temperature from 2 to 8 degrees for up to 96 hours. If you'd like to monitor the temperature fluctuations during the shipping process, we're happy to add a temperature logger in the shipment. And in the case that you purchase both collections for GCSF and combo, the packs will be shipped after each collection to ensure that you get the freshest quality upon delivery. The shipping courier that is used to ship this product has great expertise in handling time-critical and temperature-sensitive deliveries. We also may be able to provide same-day delivery in the Bay Area and the greater Boston area upon request. I've also linked the detailed terms and conditions for more information. Now that you have the product, let's explore the applications and how it can be used to advance your research. Because mobilized leukoplaques can be a source of both CD34 positive cells and all the other immune cells, they can have a wide range of applications. Starting off with the main application, which is cell and gene therapies. Gene-edited CD34 positive cells can be used to treat a range of blood disorders, such as primary immunodeficiencies and hemoglobinopathies, such as sickle cell disease, as well as metabolic diseases. We can also get immune cells derived from CD34 positive cells that can be genetically engineered with CARs or TCRs to target tumors-associated antigens or viral antigens, allowing these cells to be differentiated into T cells and B cells for enhanced immune responses in the patient. Moving on to immunotherapy. Immunotherapy can be done using the CD34 positive derived immune cells or primary immune cells isolated directly from the leukopak. These can include applications such as some of the cell and gene therapies, but also other broad immunology research. Next, we have regenerative medicine. Within regenerative medicine, CD34 positive cells can help with angiogenesis and tissue repair, promoting blood vessel formation in ischemic conditions such as peripheral artery disease or heart disease, and supporting wound healing for chronic conditions such as diabetic ulcers by enhancing blood flow and tissue regulation. Finally, last but not least, we have basic research. CD34 positive cells have many applications, such as generating humanized mouse models, studying hematopoiesis and regulation of stem cell self-renewal and differentiation for balance, studying immune cell development and function, disease modeling for some of the diseases mentioned, and also drug screening and toxicology for safety and efficacy studies on hematopoietic function and immune cell development. Overall, mobilized leukopaks are revolutionizing medicine and offer many different research areas, including cell and gene therapies, immunotherapy, regenerative medicine, and basic research. Now I'll pass it back to Annie, who will discuss the scientific overview of these mobilizing agents, and then she'll go into some of the scientific protocols involved in leukopak processing, isolating, and gene editing.
Thank you, Saba, for sharing an overview of our product offerings. Now, let's dive into what mobilization is and how it works. In normal donors without mobilization, CD34 positive cells remain in the bone marrow niche. This is due to the complex interplay of adhesion molecules and cellular interactions that regulate their localization, survival, and quiescence. A critical interaction has been demonstrated, for example, between the CXCR4 receptor and stromal-derived factor SDF1 alpha. SDF1 alpha is a chemokine protein constitutively re-expressed on various stromal cell types. The chemokine receptor CXCR4 is expressed on CD34 positive cells. These interactions resulted in approximately 3.3% CD34 positive cells in the bone marrow and very few CD34 positive cells in the peripheral blood. This makes peripheral blood a very inefficient source for isolating CD34 positive cells. However, with the use of mobilizing agents, CD34 positive cells can be moved into the peripheral blood from bone marrow. One such agent is GCSF, granulocyte colony stimulating factor, which is a growth factor that works to promote bone marrow proliferation and stimulate neutrophil production. The proteolytic enzymes produced by neutrophils reduce SDF1 alpha and CXCR4 expression, which disrupt their interaction. This works synergically with other integrins and chemokines to cause a cascading effect to loosen the anchor that keeps CD34 positive cells in the bone marrow niche, allowing CD34 positive cells to migrate into the bloodstream. Another agent is plerixafor, which reversibly inhibits the binding of CXCR4 to SDF1 alpha in a concentration-dependent manner. This in turn releases CD34 positive cells into the circulation. The combo drug, which is a combination of GCSF and plerixafor, utilizes the mechanisms of both drugs to allow more CD34 positive cells to flow into the bloodstream. In summary, GCSF and plerixafor utilize different mechanisms to mobilize CD34 positive cells from the bone marrow to the bloodstream. So, how does a researcher decide which regimen to use? The choice depends on the specific research goals, whether the priority is maximizing cell yield or avoiding certain biological effects. To help with this decision, let's take a closer look at the key features of each regimen. GCSF is the most commonly used regimen in clinical practice, whereas plerixafor is favored by some researchers because it does not stimulate the bone marrow, resulting in a cleaner product with fewer granulocyte contamination. In addition, in a clinical setting, plerixafor is chosen for patients with certain diseases such as sickle cell anemia, as using GCSF may lead to life-threatening side effects. If researchers want to use mobilized leukopak from normal donors as a control, they may then elect plerixafor regimen. Additionally, the cell yield varies depending on the regimen used. The combination drug produces the highest number of total nucleated cells and the frequency of CD34 positive cells, while plerixafor uses a relatively lower amount and GCSF falling in between. Depending on the researcher's specific needs, the appropriate regimen can be selected. Once the regimen is picked, we will schedule the collection and deliver the mobilized leukopak to you. Upon receipt of the mobilized leukopak, you may proceed to the isolation of CD34 positive cells. Here, we want to briefly walk you through our isolation process using EasySep kits. I also want to share with you what we can support for one of the downstream applications, gene editing. When isolating CD34 positive cells, we recommend using our EZ250 magnet paired with the EasySep CD34 positive cell isolation kit. The EZ250 magnet can fit a T75 flask and can process up to 12 billion cells at a time to increase isolation efficiency. Prior to isolation, we want to first process the mobilized leukopak. For mobilized leukopak larger than 150 ml, start by concentrating the cells by centrifugation at 300 G for 10 minutes. After aspirating the supernatant, resuspend the pellet in recommended media, which is Ribosep buffer 2 or PBS with 0.5% BSA and 2 millimolar EDTA without calcium or magnesium. Then proceed to red blood cell slices by mixing the samples with ammonium chloride and incubating on ice for 15 minutes. For mobilized leukopak smaller than 150 ml, you may proceed to red blood cell slices directly without concentrating the sample. Afterwards, spin the sample down at 300 G for 10 minutes and top up the tube with recommended media after aspirating the supernatant. Then, we want to reduce the platelet contamination by doing a slow spin at 120 G for 10 minutes with break off. Finally, after resuspending the cells, we can proceed to CD34 positive cells isolation. For this kit, we recommend a resuspending concentration between 100 to 200 million cells per ml. If you go with a higher concentration, you may see enhanced purity, while the lower concentration may improve recovery. You may choose the concentration that best suits your need. For CD34 positive cells isolation, start by adding the EasySep isolation cocktail to the sample in a T75 flask and mix thoroughly. Incubate at room temperature for 10 minutes. Next, add the vortex rapisphere into the cells and incubate for 5 minutes. Then double the sample volume with the recommended media that was used in the previous processing step. And carefully place the T75 flask containing the cells in the EZ250 magnet. We recommend using the easy-to-fit magnet as it can process much larger amount of cells compared to smaller magnets to increase isolation efficiency and reduce cell handling time. After a 10-minute incubation in the magnet, carefully pipette off the supernatant. Then remove the flask containing the isolated cells from the magnet and top up to double the original volume with the recommended media. Given the high concentration of CD34 positive cells, after the first separation, the sample can be passed through a 70-micron strainer to remove aggregates if present, to obtain optimal purity and recovery. Then you will repeat the magnetic separation steps 2 more times. If necessary, you may add additional separation, which may improve purity but may reduce yield. After the last separation step, take the T75 flask out of the magnet and resuspend the cells. At every resuspension step, be sure to collect the cells from the sides of the flask by rinsing them with media. After carefully pipetting the cell suspension into a new tube or a centrifuge bottle, you may centrifuge the sample at 300 G at 10 minutes with brake low to wash the cells one last time. Then you may aspirate and discard the supernatant and resuspend the cells to desired cell concentration. Now the cells are ready to use for downstream applications. If this is your first time performing mobilized leukopak isolation, our reps are happy to come assist you in the lab. The total processing time for CD34 positive cells isolation can take around 3 hours, depending on the cell count, which is quite efficient considering the amount of cells being processed. If more than one easy-to-50 magnets are used, and if more than one person is performing the isolation, this process can take significantly shorter. Here is a representative flow cytometry data before and after CD34 positive cells isolation using the method described. The purity of CD34 positive cells in the isolated fraction is typically 94.6 plus minus 2.4%. As positive selection is used to isolate these cells, we recommend using anti-human CD34 antibody clone 581 or clone 8G12, which we offer. After isolation, you can use our STEMCELL media and supplements for culture, expansion, and differentiation, which Saba will cover in more detail shortly. For the negative fraction of the mobilized leukopak after CD34 cells isolation, you may use our EasySep kits to isolate immune cells, such as different manners of T cells, monocytes, NK cells, B cells, as well as PBMCs, etc. We offer 10x size kits, so there is no need to combine the reagents yourself. For some of these cell types, we offer both positive and negative selection kits to meet your specific requirements. Once isolated, you may care for them using our immunocal media and supplements, which Saba will introduce in more detail as well. If you're looking to outsource the mobilized leukopak isolation, we provide GCSF mobilized leukopak bulk isolation service, and we can provide the cells directly to you. We'll also be able to offer the same service for Plerixifer and combo mobilized leukopaks in the future. Now, let's shift our focus to a different topic, gene editing. Gene editing is a common application of mobilized CD34 positive cells, which can be achieved by a recently launched instrument, CellPore. CellPore utilizes mechanoporation to achieve efficient and gentle cargo delivery into the cytosol of the cells. Well, how does this instrument work? Let's take a closer look. First, mix the CD34 positive cells with a cargo, in this case, CRISPR-Cas9 RNP, and load the mixture in the delivery cartridge, which is the size of an Eppendorf tube. The instrument adds air pressure to the cartridge and pushes the cells and cargo mixture through the microfluidic chip. As the cells flow through the chip, the cell membrane is temporarily disrupted to facilitate intracellular delivery of the CRISPR RNPs directly into the cytosol. Then the cells are collected in the bottom of the collection tube and are transfected. This process takes about 3 seconds. In addition to CRISPR-RNP, the CellPort instrument can also be used to deliver small molecules, peptides, RNA, and antibodies. Aside from transfecting in CD34 positive cells, they can also be used in immune cells such as different manners of inactivated T cells, NK cells, and PBNCs. If you are interested in this instrument, we're happy to share more information about this. Now, I will hand it back to Saba to discuss more downstream products.
Thank you, Annie, for walking through the isolation and gene editing workflows. I will continue on with the culture, expansion, and differentiation media workflows, along with analysis and cryopreservation workflows shown here. Mobilized Leukopaks can generate a large number of CD34 positive cells for your experiments. Whether you wish to culture and maintain the stemness of these CD34 cells or expand them to make more, or stem cell product line provides the optimal solution. We offer a wide range of STEMCELL media specifically designed for CD34 positive cell expansion, catering to our diverse research needs. All of these formulations are serum-free, including STEMSPEN, SPEM, SPEM2, and XF, which stands for xeno-free. And then we also have select options that are CGMP grade, including STEMSPEN EOF, which stands for animal origin-free, for enhanced consistency and regulatory compatibility. STEMSPEN media do not contain any cytokines, which gives you the freedom to customize your culture conditions by adding STEMSPEN expansion supplements or your own combination of cytokines and additives. These products are compatible not only with Mobilized Leukopaks, but with all of our CD34 positive tissue sources, including cord blood and bone marrow. In the next slide, I'll show how all 4 versions of STEMSPEN support the expansion of mobilized peripheral blood CD34 positive cells and compared to our competitors. This figure presents the expansion data of CD34 positive cells isolated from Mobilized Leukopaks and cultured in all 4 STEMSPEN medias and comparing them to commercial alternatives. The purified CD34 positive cells were taken from GCSF Mobilized Leukopaks and cultured in STEMSPEN media, which are shown in the 4 orange bars, and then 6 competitor media shown in the gray bars. These were also added with the STEMSPEN CD34 expansion supplement and 1 micromolar of UM729. After 7 days, flow cytometry analysis confirmed that STEMSPEN media supported similar or greater expansion of CD34 positive in graph A, and also CD34 positive, CD45 RA negative, and CD90 positive markers, which are key indicators of primitive hematopoietic stem and progenitor cells. This demonstrates that STEMSPEN provides an optimal environment for ex vivo hematopoietic stem and progenitor cell expansion. Additional data was collected for other markers such as CD133 positive, EPCR positive, and really showed comparable results. And fold expansion was also normalized to STEMSPEN FEM, and all of this data is available on our website. In this study, frozen mobilized peripheral blood CD34s were thawed before use, but we expect that freshly isolated CD34 positive cells from Mobilized Leukopaks will perform equivalently, if not better. Beyond maintaining stemness, the STEMSPEN portfolio also supports the differentiation of CD34 positive cells into various lineages, and we'll explore that in the next slide. So after you have enough CD34 positive cells and your goal is to differentiate these into various cell types, whether it is the myeloid lineages or the lymphoid lineages, we have STEMSPEN differentiation products that can help you with that. We have many different STEMSPEN reagents, including both media and supplements that can come as a kit or standalone products for you to mix and match for your research needs. And if you need help determining which media formulation to use, we have a dedicated customer support team that can help you design your optimal media condition as scientists supporting scientists. If your starting source was CD34 positive cells, then I suggested the STEMSPEN media portfolio. However, if your starting source is the isolated immune subsets directly from the Mobilized Leukopaks, and you'd also like to culture, expand, and differentiate them to more specialized immune subtypes, then I would direct you to the immunocult media portfolio. These are great for expanding both the myeloid cell types and the lymphoid cell types and can allow you to get more cells for your downstream experiments. These can also be a great basal medias for general culture and maintenance needs. After you have cultured your CD34 positive cells, there are many downstream experiments that you can use them for. Annie walked you through some cool gene editing technologies using CellPore. And one of the important assays for assessing the functionality of CD34 positive cells is the colony forming unit, or the CFU assay. Due to the utility of the CFU assay in evaluating the potency of hematopoietic stem and progenitor cells, it is actively being used in the clinic. Among the currently approved FDA hematopoietic cell and gene therapy products, the majority, if not all, reference and use the CFU assay. In order to perform this, we have the MethoCult product media and then the STEM Vision instrument, which is backed by over 3 decades of experience in the space. We also provide this assay as a custom service through our custom assay services division if you prefer to outsource this assay. In addition, you can further characterize the cells using flow cytometry to assess progenitor cell populations, including the frequency of those more primitive subsets I mentioned, along with the overall CD34 positive frequency at any stage of your experiments. When you order fresh mobilized leukopaks, we provide the CD34 positive frequency and major immune cell types, but if you're interested in adding these primitive cell types, you can request a custom flow panel at the time of order placement. CD34 positive cells can also be used for humanized mouse model experiments for further functionality and research, and they have many other applications in the research fields of hematopoietic stem cell gene-based therapies, regenerative medicine, transplant therapy, immunotherapy, and basic research. Lastly, if your mobilized leukopaks yield more cells that you needed for your fresh applications, especially after you expanded and cultured them, you can freeze the excess for future use. For hematopoietic stem cells or CD34 positive cells, or recommended freezing media is STEMSpan SPEM with 10% DMSO. This is what is used internally for manufacturing or own frozen isolated subsets of CD34 positive cells from all of the three different tissue sources. In addition, CryoStor CS10, which is a GMP grade alternative, may also be used for CD34 positive cells. But CS10 is our standard freezing media used for majority of our primary cell types outside of CD34 positive cells, and we recommend it for those isolating their own frozen immune cells. After you've frozen these cells can then be thawed to ready to go for experiments as you need them. And now this concludes the section of all of our downstream products and workflows. So why use fresh mobilized leukopaks from STEMCELL Technologies? First of all, these are products that have really high CD34 positive cells. So this allows you to obtain these rare populations of CD34 positive hematopoietic stem and progenitor cells from a single donor in large quantities, and ensuring consistency across your experiments and enabling scale-up. Our products are also freshly collected and ethically sourced using IRB-approved protocols, giving you confidence in quality and compliance. You can also customize your products, I mentioned, by adding additional solar panels, but you can also request donor-specific criteria, additional characterization, and collection volumes tailored to your specific research needs. By obtaining high yield and quantity of CD34 positive cells from a single product and a single donor, you can really be efficient with your time and accelerate stem cell research, gene editing, and cell therapy development. Not only do we provide high yield, freshly collected, and ethically sourced mobilized leukopaks, but we also offer all the downstream products you need to maximize their potential. From isolation to expansion to differentiation and analysis, we support your entire workflow, helping you achieve your results faster and more efficiently. With STEMCELL Technologies, you get more than just the cells, you get a full solution. And not only do we provide the cells, but we also provide many different sources to use those cells. So we have a lot of sources of information that are shown here and linked here as well. We have overview pages, a frequently asked questions page, an overview of the comparison of those hematopoietic CD34 positive cell sources, and many different flyers that show our full workflow. In addition, we also have a lot of technical resources such as a leukopak processing webinar, and we walk through how the nuances between a mobilized leukopak processing in this presentation. We also have a cell frequency wall chart to get all of different frequencies from the different sources. And in addition, we also have workflow resources. So I've linked all of our brand pages for isolation, differentiation, gene editing, and many more. And we're also always building on this content. You can also contact us for any primary cell needs and contact our customer support for any experimental design. With that, I would like to conclude the webinar and really hope you enjoyed it from both me and Annie. Annie's email is also listed here. Thank you so much for joining, and we will take any questions.
Excellent. Thanks so much, Saba and Annie, for the great presentations. And with that, move right on to the Q&A. And so I'll invite Annie and Saba back onto the audio line here. All right, Saba and Annie, ready to answer a few questions? Awesome. So our first question is, how long does CD34 positive cell isolation from a mobilized leukopak take?
Thank you, Liam. I'll take this one. The time to spend to isolate CD34 positive cells using our EasySep kit depends on the cell count, the final total nucleated cell count. The more cells there are, the longer it may take. For example, a GCSF mobilized leukopak, we would expect about 3 hours. That's from our in-house data with EZ250 magnet, assuming we have one person processing and doing the isolation with the one EZ250 magnet. Given the high cell count in mobilized leukopak, you may need to split the leukopak into 2 halves and perform the isolations on each half sequentially due to the volume limit of the magnet. Specifically, if I say I have a 40 billion cells and resuspend them at 200 million cells per mil, I would end up with 200 mil. And since our magnet has a volume limit of 125 mil, I would split them to 100 mils each and do the isolation on the first 100 mil, then the second 100 mil. But if, say, I have a second magnet, I can do the isolations on each half, like side by side. So, which in that case, I would be cutting the isolation part of the process by half the time spent, which can be about 1 hour, depending on this total cell count. And you may be able to finish the whole process in around 2 hours.
Excellent. Yeah, some great tips there. All right, next question here. Will I be able to isolate both CD34 positive cells and T cells from the same mobilized leukopak?
I'll take this one as well. Yes, you will be able to. Depending on the amount of CD34 positive cells you're looking for, you can either do a sequential isolation or split the packs. Specifically, say you could first use our positive selection kit to isolate CD34 positive cells, then isolate T cells from the negative fraction using either positive or negative selection method of your choice. Or you can split the mobilized leukopak and isolate CD34 positive cells from half of the pack and T cells from the other half.
Excellent. All right, question here. Can I purchase one of the 2 mobilized leukopak collections?
Yes, I can take this question. So you may purchase either the first collection on day 5 or the second collection on day 6 for the GCSF or the combination mobilized leukopaks. And you may also purchase both of the packs if you wish. These packs will be shipped on the day that they are collected, so you'll get the freshest product available. So please plan your experiments accordingly, whether it's overnight shipping or same-day shipping. In the case of Plexifor, it's just 1 pack, so you will get that the same day it's collected as well.
Excellent. Thanks a lot. All right, this is a question about delivery. This person writes that they're in the San Francisco or the San Francisco area, but maybe you can talk about different shipping delivery. But when would same-day delivery arrive for different areas around the U.S.?
I'll take this one. For San Francisco area, we'll be able to deliver mobilized leukopak on the same day of collection, and that will arrive about 3 p.m. or later, depending on traffic and your location. We also can provide same-day delivery for Boston area as well, and that would typically be about 4:30 or later, depending again on traffic.
Excellent. Thanks a lot. Okay, somebody writes, can I recall donors for mobilized leukopak or non-mobilized leukopak?
Yes, donors can be recalled for both. So for a normal non-mobilized leukopak donor, they can usually donate once every 1 to 2 months. But this is highly donor dependent and cannot be guaranteed because it's ultimately the donor's choice whether they want to be come in for another collection or not. For a mobilized leukopak, due to the nature of the mobilization, they can be mobilized 2 to 3 times in their lifetime. But the actual number of mobilizations is really donor dependent and depending on the donor site as well, with the guidance from the medical director, because each donor can react differently to the mobilizing agents. So we just want to make sure that the donor's health is of the utmost priority. It may be possible to obtain both the non-mobilized and mobilized products from the same donor, especially if you want it prior to mobilization or post-mobilization. But please discuss this with us as early as possible so we can make sure it's feasible and accommodate your request before you're placing your order.
Excellent. Thanks a lot. Well, I think that's all the questions we have time for. So Saba and Annie, thanks so much for being with us today. It's been a really pleasure having you here.
Thank you.
Thank you so much.
Thank you.
Also, big thanks to the audience for your time today. And in closing, we hope you enjoyed the Scientist.com and STEMCELL Technologies webinar, and we'll see you again next time. Have a great day, everyone.